In this chapter, we'll be taking a look into human development. We'll begin by looking at fertilization and the pre-embryonic stage, as well as the embryonic and fetal stages. Then we'll consider some adaptations in the neonate or the newborn, as well as aging and senescence. A miraculous aspect of human life is the transformation of a one-celled fertilized egg into an independent, fully developed individual. It's quite an amazing feat. Embryology is the study of all of the prenatal development. And developmental biology is a broader science that embraces change in form and function from the fertilized egg through old age. Now, the term embryo can be confusing between different authorities. Some say that an embryo are the stages that begin with the fertilized egg, or the two-cell stage produced by the very first cell division. Other authorities and others consider that it's only at 16 days when the individual consists of 3 primary germ layers that we can call that individual an embryo. The 3 primary germ layers are ectoderm, mesoderm, and endoderm. Embryogenesis are the events that lead up to this stage. And pre-embryonic stage are the first days after fertilization. This second way is the way that we'll consider the term in this class. If it's to survive, an egg needs to be fertilized within 12 to 24 hours of ovulation. That means the sperm need to encounter the egg somewhere in the distal 1/3 of the uterine tube, because it takes about 72 hours for the egg to reach the uterus. The vast majority of sperm don't make it to the egg. A lot of them are destroyed by the vaginal acid or drain out of the vagina. Some of them fail to penetrate the mucus of the cervical canal. It's quite thick and there's a plug there. Others, once they do enter the uterus, are destroyed by leukocytes. And of those, half of them go up the wrong tube. So of the 300 million sperm that were ejaculated in a healthy ejaculate, only about 2,000 to 3,000 will actually reach the vicinity of the egg, and only one of those will be lucky enough to fertilize the egg itself. Sperm move by lashing the tail as they crawl along the female mucosa. They can be assisted by a number of different things in female physiology. First of all, in the cervix, there are strands of cervical mucus that can guide the sperm through the cervical canal. Also, during orgasm, a female has uterine contractions, and those contractions can literally suck the semen from the vagina and spread it throughout the uterus. There's also some evidence that there are chemical attractant molecules that are released by the egg that attract the sperm from a short distance away. Now, before the spermatozoa reach the uterine tube, within about 5 to 10 minutes of ejaculation, they need to undergo some modifications so that they can fertilize the egg. This process is called capacitation. It's a process that migrating sperm must undergo to make it possible to penetrate the egg. The plasma membrane of the fresh sperm is toughened by cholesterol. This prevents premature release of the acrosomal enzymes in the head of the sperm while it's still in the male reproductive tract. If it were to be released in the male reproductive tract, these enzymes could cause damage to the sperm ducts. So once the sperm enter the female fluids, they're able to leach the cholesterol from the sperm's plasma membrane and dilute the inhibitory factors that are in semen that prevent its acrosome from bursting while it's in the male reproductive tract. Because of the removal of the cholesterol, the sperm membrane becomes very fragile and permeable to calcium. As the calcium diffuses into the sperm, it causes more powerful lashing of the tail, so it actually swims faster towards the egg because of the exposure to female fluids. So even though sperm might reach the egg within about 5 to 10 minutes of ejaculation, it takes about 10 hours for the process of capacitation to occur. Now, sperm can remain viable for up to 6 days after ejaculation. And as we've already discussed, the egg needs to be fertilized within about 12 to 24 hours of ovulation. So there's quite a broad window in which fertilization could occur. Pregnancy is fairly unlikely from intercourse that occurred more than a week before ovulation, and it's also unlikely, considering the process of capacitation as well as the journey itself, that an egg would become fertilized beyond 14 hours after ovulation. So conception is optimal if the sperm are deposited a few days before ovulation or up to 14 hours after. Now, there are more modifications that the sperm needs to have happen in order for it to actually fertilize the egg. When it encounters the egg, it undergoes an acrosomal reaction. That is exocytosis of the acrosome. Now, the acrosome is the very tip of the sperm's head, and it contains a number of enzymes that help it penetrate the egg. So in all truth, it doesn't take only one sperm to penetrate an egg. It takes the enzyme of many sperm to be released in order to clear the path for the one that will actually get to penetrate the egg. The sperm not only need to penetrate the egg cell membrane, but all the layers that surround it that you might remember from the female reproductive chapter. There's a zona pellucida and a zona granulosa. So there's all these granulosa cells that are tightly packed and attached to each other that need to be moved apart from each other so that sperm can actually penetrate even the zona pellucida. So there are two primary acrosomal enzymes. Hyaluronidase is the first one. It digests the hyaluronic acid that binds the granulosa cells to each other. The other is acrosin. Acrosin is a protease, an enzyme similar to trypsin that we find in the digestive system that digests this zona pellucida so that the sperm can finally touch the cell membrane. When the path has been cleared by many sperm, one of them will bind to the zona pellucida and release its enzymes. This digests the path through the zona, and then it will contact the egg itself. The sperm head and the midpiece enter the egg, while the tail is left behind. Now the egg destroys the sperm and the mitochondria, and thus only maternal mitochondria are passed on to an offspring, because the paternal mitochondria have been destroyed by enzymes in the egg. If two sperm were to penetrate the egg, we'd have a lot of problems. That would be called polyspermy. And in the case of polyspermy, we now would have the DNA that's in the egg, a haploid set of DNA from the egg, and a haploid set of DNA from each sperm that entered. So rather than have a diploid or a 2N individual, there would be 3N or 3 sets of chromosomes. And that simply won't work. That egg is not viable, and so would die almost instantly. There are two mechanisms, though, that prevent polyspermy from happening. The first is a fast block system. This is where, when the sperm binds to the egg, it opens a bunch of sodium channels in the egg membrane. As sodium flows into the cell, it acts much like a neuron and depolarizes the membrane, and that inhibits the attachment of any more sperm to the egg. The second mechanism is a slow block. This involves the secretory granules or cortical granules just below the membrane. You can see those in green right here. In addition to allowing an inflow of sodium, when the sperm penetrates, it causes an inflow of calcium. This stimulates the cortical reaction, in which the cortical granules literally release their secretions beneath the zona pellucida. This secretion swells with water and pushes any remaining sperm away from the egg and creates an impenetrable fertilization membrane between the egg and the zona pellucida, so no other sperm can enter the egg. So that is, there are two mechanisms to prevent polyspermy, that is, more than one sperm entering the egg. Now is a great time to take a moment and diagram fertilization. This figure below might serve as a good guide. Think about the acrosomal reaction and the cortical reaction and how the sperm actually ends up penetrating the egg membrane. And then how are other sperm blocked from fertilizing the egg? So after the egg is fertilized in the distal 1/3 of the oviduct, it'll undergo meiosis II. And as it journeys through the oviduct towards the uterus, it'll continue in cell divisions to form a blastocyst, which floats freely in the uterus until it implants at about 6 days after fertilization. The secondary oocyte, that is, it's gone through meiosis I, it begins meiosis II shortly before ovulation and completes it only if the egg is fertilized. So that means all of the eggs that a woman has stored in her ovaries are arrested after meiosis I. As they mature in the follicle, they begin the process of meiosis II. However, it won't complete. Cytokinesis will not occur until the egg is fertilized. The sperm and the egg nuclei are now both inside the same cell. They will swell and become what are called pronuclei. Each pronuclei will rupture, and the chromosomes of the two gametes will mix to form a single diploid set. That is, we've restored the 2N condition in this zygote. The fertilized egg is now called a zygote. It's ready for its first mitotic division. So remember, meiosis was for the formation of gametes. Mitosis is regular cell division for the development of tissues. There are major stages in prenatal development. As we're all fairly familiar, the course of pregnancy is divided into 3-month intervals called trimesters. The first trimester lasts through the first 12 weeks from fertilization. More than half of all embryos actually die in the first trimester. The conceptus is most vulnerable to stress, drugs, and nutritional deficiencies. During this time, there's also some checks and balances system that makes sure that the embryo is a healthy embryo. So most embryos with genetic abnormalities are spontaneously aborted during the first trimester. The second trimester lasts from weeks 13 through 24. The organs complete most of their development during this phase, and the fetus looks distinctly human in its shape. And there's a good chance of survival if the fetus is born near the end of the second trimester. The third trimester lasts from 25 weeks to birth at 40 weeks. The fetus grows rapidly and the organs achieve enough cellular differentiation to support life outside the womb. At 35 weeks, the 5.5-pound fetus is considered mature. Before 35 weeks, it's premature. Now let's take a look at the biological stages of development. The preembryonic stage, as I already introduced, is the first 16 days of development. It culminates in the existence of an embryo, and it involves these 3 major processes that we'll consider shortly: cleavage, implantation, and embryogenesis. The next stage is the embryonic stage, and following the embryonic stage, we'll have the fetal stage. In the preembryonic stage, the zygote is on its journey down the oviduct into the uterus, and it begins its implantation. Cleavage is where we see mitotic divisions occurring in the first 3 days while the conceptus migrates down the uterine tube. The first cleavage occurs within 30 hours after fertilization. The zygote splits into 2 daughter cells. Each of these daughter cells is called a blastomere. By the time the conceptus arrives in the uterus, at about 72 hours after ovulation, these 2 daughter cells have further divided into 16 cells. We call this the morula stage because it's a hollow ball of cells and it resembles a mulberry, sort of bumpy all over the surface. It's still no larger than the zygote itself, just each cell has divided and divided and divided and become smaller. So cleavage then produces smaller and smaller blastomeres as each blastomere divides to form more. The morula will lie freely in the uterine cavity for 4 or 5 days, and it can divide into 100 cells or so. The zona pellucida that surrounded the original egg membrane disintegrates and releases the conceptus, or the blastocyst. The blastocyst is a hollow sphere with an inner cell mass. The trophoblast is the outer layer of squamous-shaped cells. These are destined to form the placenta, and that will play a role in the nourishment of the embryo. So inside the ball is a hollow space and a mass of cells. The mass of cells is called the embryoblast, and the hollow space is called the blastocele. The embryoblast is the portion of the blastocyst that is destined to become the embryo. Okay, take a moment now just to draw a blastocyst, including the blastocele, the trophoblast, and the embryoblast. So here's a question. What actually produces twins? We're all probably aware that there are 2 types of twins. There are identical twins and those that are non-identical. The dizygotic twins account for about 2/3 of twins. These are ones that are from 2 zygotes, dizygotic, so they are non-identical twins. 2 eggs are ovulated and 2 eggs are fertilized, forming 2 zygotes and usually 2 placentas. These zygotes are no more or less genetically similar than any other siblings, and they implant separately into the uterine wall. Monozygotic twins, on the other hand, result from 1 fertilized egg, or 1 zygote, but the embryoblast later divides into 2. We're unsure why, but these individuals are genetically identical, and they're of the same sex and nearly identical in appearance. Now, there's some question as to whether they truly are genetically identical. One of the theories about why the embryoblast divides is that some of the cells have a mutation in them that appear different than the other cells. So the embryoblast rejects the cells that are different than itself, and that actually becomes a whole new individual. But they're similar enough to call them identical for our purposes. So now that we have our blastocyst floating around in the uterine cavity, how does it actually implant into the endometrium? The blastocyst, after it's been released from the zona pellucida, will implant in the wall after about 6 days after ovulation, usually on the fundus of the posterior wall of the uterus. Implantation is the process of attachment to the uterine wall, and it begins when the blastocyst first adheres to the endometrium. The trophoblast cells are the first to adhere. They'll separate into 2 separate layers. There'll be a superficial layer, which is in contact with the endometrium, and a deep layer, which is closer to the embryoblast. The plasma membrane will break down in the superficial layer, and the trophoblastic cells will fuse into a multinucleate mass called the syncytioblast. You can see that here illustrated as the blue cells. The deeper layer is going to be closer to the embryoblast. This portion is called the cytotrophoblast. And these cells retain their normal appearance and have membranes that divide them from each other. The syncytiotrophoblast will grow into the uterus like little roots. They digest endometrial cells along the way, and the endometrium reacts to this injury by growing over the blastocyst and covering it. The conceptus becomes completely buried in endometrial tissue. The process of implantation takes about a week, and it's completed about the time the next menstrual period would have started had the woman not become pregnant. The trophoblast also secretes a hormone called human chorionic gonadotropin, or HCG. This is the hormone we measure to determine if a woman is pregnant or not. HCG stimulates the corpus luteum to secrete estrogen and progesterone. Remember, the corpus luteum is the place where the egg was ovulated from on the ovary. And the progesterone that's secreted will suppress menstruation. Human chorionic gonadotropin levels rise in the mother's blood until the end of the 2nd month of pregnancy. The trophoblast develops into a membrane that's called the chorion, and it takes over the role of the corpus luteum and thus makes HCG unnecessary, because HCG was stimulating the corpus luteum to release estrogen and progesterone. The ovaries will become inactive for the remainder of pregnancy, and estrogen and progesterone levels will rise because of the secretions from the chorion. So again, a good time to take a moment to draw a quick diagram. Figure out the relationships between the cytotrophoblast and the syncytiotrophoblast and how these guys make their way into the endometrium. Quick picture. It's important to take the time to do these while the information is fresh in your head. Now, if the egg doesn't implant in the uterine wall but implants somewhere else, it's called an ectopic pregnancy. About 1 out of 300 pregnancies are ectopic. Some of them are tubal pregnancies. That is, they implant in the uterine tube. For some reason, the egg gets stuck. It's usually due to an obstruction, such as a constriction that results from pelvic inflammatory disease or a tubular surgery, maybe a previous ectopic pregnancy, or repeated miscarriages. Now, the tube, because of the growing embryo, will rupture within about 12 weeks. It's extremely painful and very dangerous. About 1% of ectopic implantations occur in the abdominal pelvic cavity. That's about 1 out of 7,000 fertilizations. This can really threaten a mother's life also. 9% can result in live birth by cesarean section, which is kind of a wild concept. Basically, in this case, the egg doesn't make it into the fallopian tube, and it gets fertilized and is just released into the abdominal cavity because the ovary is not actually attached to the oviduct. There are fimbriae that wave and pull the egg in. So once the embryo is implanted, what happens? This is when we reach the stage of embryogenesis. We have arrangement of blastomeres into 3 primary germ layers. We're forming the actual embryo. These 3 layers are the ectoderm, mesoderm, and endoderm. The embryoblast will separate slightly from the trophoblast. This will create a narrow space between them, which will become the amniotic cavity. The embryoblast flattens out into an embryonic disc, which is initially just composed of these 2 layers. The epiblast, which faces the amniotic cavity, and the hypoblast, which faces away. The hypoblast cells will multiply to form the yolk sac, which will nourish the embryo before the placenta has formed. The embryonic disc is flanked by 2 spaces, the amniotic cavity on one side and the yolk sac on the other. Once the embryonic disc has formed, a primitive streak forms along the midline of the epiblast. This is just a thickened cell layer. Down the middle of the primitive streak will be a groove called the primitive groove. These events are the events that make the embryo bilaterally symmetrical, divided into even halves. The multiplying epiblast cells will migrate medially towards the primitive groove and down into it. They replace the original hypoblast with a layer called the endoderm. So these red cells will fill in the space here. This is the process of gastrulation. A day later, these migrating epiblast cells will form the layer that we call the mesoderm. The remaining epiblast is called the ectoderm. So now we have our 3 germ layers. The epiblast has become the ectoderm. The cells that migrated down through have become the mesoderm, and the endoderm is beneath that. The mesoderm is more loosely organized than either of the other layers, and it'll differentiate into a loose fetal connective tissue called mesenchyme. Mesenchyme will give rise to muscles, bones, and blood. It's composed of a loose network of wispy mesenchymal cells that are embedded in a gelatinous ground substance. Once these 3 primary germ layers have formed, embryogenesis is complete, and we have what's considered to be the embryo. It's 2 millimeters long and about 16 days old. So, that was a lot of information in the past few slides on embryogenesis. Absolutely, take a moment here to outline the process of embryogenesis. What goes on? How do we get the 3 different germ layers? Ectoderm, endoderm, and mesoderm. Remember that endo means inside, ecto means outside, and meso means in the middle. You can see in the figure down here how these layers arrange themselves also. Just gives you another perspective.